had time to work on your membrane lab and you finished your organelle quiz. A couple of reminders before we get started. 15 and 16 homework is due tonight. Well, that's not supposed to be there. Quiz 1 you took today. 15 and 16 you do have a quiz on Thursday. And you have another quiz on the cell membrane on Friday, but be prepared. That quiz will have questions about organelles and enzymes as well, because we have to know all of that. We can't forget any of it. So today we're going to talk about the membrane. We're going to do our little bubble lab. But let's talk about the things you read and figured out real quick. Maybe. All right, here we go. So you read about what it told you about a membrane, and it told you that the membrane was a bilayer, and it was made up of phospholipids. And in those phospholipids, the phospholipids have two different properties. There's hydrophilic and hydrophobic. And it asks you to, number one, asks you to label the phospholipid molecule as which part is hydrophilic, which part is hydrophobic, but you had to figure that out. So let's look at the bilayer first and pretend like it's a cell. And let's ask ourselves, if this was a cell, what would be outside of the cell? Now, you better understand cells in our body that make up our tissues, they are surrounded by fluids, and those fluids are mostly water. And inside the cell is filled with that jelly-like substance called cytoplasm, but cytoplasm is mostly water. So based on that, can you figure out which part of the cell membrane is which part of the phospholipid is hydrophilic and hydrophobic? To do that, let's make sure we understand what the words mean. Hydrophobic means water-fearing. So it repels water. And hydrophilic means it's water-loving, or that it is attracted to water. So if we look at this diagram of where the water would be, it would make sense to say that these purple circles, or the phosphate heads, is hydrophilic, because it is positioned itself to where it is always facing the watery environment. And these tails, they're called fatty acid tails. This is where we get the lipid part of the name. The fatty acid tails are stuck in the middle. They're away from the water. And so if they're away from the water, they're not wanting to be by it, they're going to be hydrophobic. And if you think about it, if you're afraid of something, like picture yourself in a haunted house and some big scary clown jumps out, you're going to run away from the thing that scares you. So if you look at this phospholipid, these heads like water, but the tails don't, so they are pulling away from the water. Well, these tails are pulling away from that water on the inside, and so they're pulling towards each other, and that's what holds that bilayer together. That attraction or the repulsion from water. It's called a hydrophobic interaction. Now, question 2 just asked us to label where the water was, and we've already done that. We're kind of just going a little out of order, but that's okay. The next question said, in your own words, explain why the phospholipid bilayer self-assembles. Well, we just talked about that, but it self-assembles because the fatty acid tails repel water and pull together to avoid watery environments. This leaves the hydrophilic heads. I'm sorry, guys, it's been a long day. Hydrophilic heads exposed to the water. All right, and we'll learn more about this as we go. What we're going to do next is we're going to actually simulate the cell membrane and learn about some of its characteristics through the bubble lab. Some characteristics that we're going to learn about through this lab is that cell membranes are flexible, they can self-repair, or they can reseal themselves if they're torn. They contain molecules other than phospholipids. They are selectively permeable. Permeable means that something can go through it. So if it's selectively permeable, it picks and chooses what can go through the membrane. And fifth, we are going to learn that membranes can split to form new cells, or to form vesicles. So that's what we're going to get out of this lab, and we should have a lot of fun playing with some bubbles while we're doing it. All right, guys, so we finished the cell membrane lab, and now we're going to watch this video on the cell membrane by the Amoeba Sisters. If you're at home, I'm not going to play this video right now, but it is linked in Google Classroom, and it's linked in the PowerPoint slides, so please watch it. It does give really good information, but I'm not going to video it playing on the screen. So go ahead and just watch that on Google Classroom. So now as we go through these, you're going to be filling out your note sheet that you picked up when you came into class. The note sheet is fill in the blank, goes right along with the slides. So just as we go through it, fill in the blanks, add to it if I add extra information. And it shouldn't take too long, but we should get lots of good information. All right, so functions of the cell membrane. The cell membrane, obviously it contains the cell contents. It holds it all together, and it separates all of those contents from the external environment that the cell is in, whatever that is. Without the membrane, there is no cell. There's nothing that separates the cell out, so it would just be a bunch of stuff floating around and just floating around, not separated out, not functional together. It also controls what enters and leaves the cell. It is selectively permeable. It also functions in cell identification. This is why when you get strep, your body actually acts sick because it recognizes those strep bacteria as foreign based on what the proteins that are in their cell membrane. This is why you can't get blood that doesn't match your blood type. If you're type A, you can't get type B blood because of the cell membrane on type B blood cells. Your body doesn't recognize them. It says, hey, bad guy, foreign invader, get out. So the proteins that are part of the membrane are how our cell body can identify our cells as normal or local cells that are supposed to be in our body, or foreign cells, invaders, cells that are not supposed to be there. So we'll talk about some of those proteins in just a little bit, but just know that those are the proteins, that is one of the functions of the cell membrane. Another function is to maintain homeostasis. And homeostasis is a stable internal environment. And as we move on with the cell membrane, we're going to learn some ways that the cell membrane functions to help maintain this. But those are the functions, some of them new, some of them are the ones you learned with the organelles. Next, the cell membrane, we call it a fluid mosaic model. Scientists like to create models and give them names to help you remember things and remember characteristics. So they call it a fluid mosaic model. And they do that for several reasons, but to understand why it's called a fluid mosaic model, you have to understand a few things about the structure. The cell membrane is a phospholipid bilayer. That's the main part of it. But adding to those phospholipids, there are cholesterol, peripheral proteins, integral proteins, and carbohydrates. And these are all examples of types of peripheral proteins, types of integral proteins. And we're going to talk more about those as we go, but we're going to fill in the model first so you can kind of understand how they're arranged and why they have the names they have. So first, here's our phospholipids. Remember, hydrophilic head, hydrophobic tails. The tails point to the middle. And then the phospholipids are in a bilayer. So we have two layers. Within that layer, we have these brown things. These brown things, those are cholesterol. They do add to the strength and rigidity of the cell membrane. We have carbohydrates. These are the green structures on here. Remember, carbohydrates are made out of sugar. So each of these is a glucose molecule or a simple sugar molecule that builds up that carbohydrate. And we also have proteins. Notice there's two types of proteins. There's a peripheral protein. And peripheral, if you think about your peripheral vision, it's the vision on the sides, the vision that you can see if you're looking forward. What you can see to the side, that's considered your peripheral vision. So peripheral protein is a protein that sits on one side or the other. So this peripheral protein is sitting here. You could have a peripheral protein that sits here, but it sits on one side or the other. Now, integral proteins are different. Integral proteins are integrated into the membrane. They can go through one side, they can go through, so they could look like this. They could look like that. They don't have to go all the way through both sides. They can go through one side or the other, but they are actually integrated into the membrane itself and not just sitting on the side. Now, another thing I want you to look at is these carbohydrates are attached to something. So when a carbohydrate is attached to a phospholipid, this is called a glycolipid. Now, glyco means sugar, so it's a sugar plus a lipid. And over here, we have a carbohydrate attached to a protein. So can you guess what that might be called? Yep, a glycoprotein, or a sugar plus a protein. Now, if you look, there's lots and lots of pieces that are pulling this together, and this is a very simple drawing of the cell membrane. But just remember that these are very, very complex. The cell membrane is very complex, and what it does is very complex. But this is just kind of an introduction to how it's structured. So if you can make sure you go over that, make sure you're familiar with what those parts and pieces are. Why do we call it a fluid mosaic? We call it a fluid mosaic because the molecules are constantly in motion. If you remember in the soap lab, you could look at it and you could see the soap swirling around in the bubble. And they were constantly in motion, just like in a fluid. If there was a bottle of water and you put a granule of pepper in it and you let it sit there, that piece of pepper would not stay in the same spot in that bottle of water. It would move around throughout that bottle of water because those molecules are in constant motion, just like all of those molecules that you saw in the picture a minute ago. They're constantly moving around. It's also flexible, just like our bubbles. When we move them up and down, those bubbles would move up and down and bend and everything else because they were flexible. The membrane changes shape as the environment dictates that it has to. That's what gives it the fluid property. Because think about water. You pour it in a water bottle, it's the shape of a water bottle. If you pour it in a bowl, it's the shape of a bowl. So fluids can change shape just like a membrane. We call it a mosaic because a mosaic, think about a stained glass window. That's a mosaic. Lots of little bitty different pieces of colored glass that come together, and when you put them together, you get this magnificent picture. That apart, all these little pieces of glass are nothing. When they come together, they're a beautiful functional picture. So if you look at that diagram, there's lots of little pieces, and separately they're nothing. But when they come together, we have this functional membrane that controls what enters and leaves the cell. It maintains homeostasis. It serves all of those purposes. So that's where the mosaic name comes from. Now I'm going to play this video. It's a short video, but just watch and pay attention. Think about the things we've talked about as you're watching. Well, I say I'm going to play it. Let's try one more time.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins, which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
All right, guys, so the phospholipids, let's revisit those for just a minute. We said that the phosphate head, was hydrophilic. That means it likes water. And if you remember when we learned about water, water was polar. Remember, it has a partial positive and a partial negative charge. So here's our water molecule, oxygen, hydrogens, positively charged, negatively charged. So it had a polar property. Well, these phosphate heads are also polar. They have a negative charge to them. But this phosphate head is attracted to the water because the negative charge of the phosphate head is attracted to the positive end of a water molecule. So polar molecules tend to attract other polar molecules, whereas the fatty acids, the lipids, remember lipids don't dissolve in water, they were nonpolar. And because they're nonpolar, we call them hydrophobic. Things that are polar, we tend to call them hydrophilic. And I know we discussed these terms already. We're just going to make sure they're drilled into your head. So this is the polar head, and this is the nonpolar tail. But this phospholipid, it has 2 personalities. It likes water and it doesn't like water, so we call it amphipathic. And amphipathic means it has both personalities. Which means it loves water and it hates water. And those properties, that amphipathic property, is why it forms a bilayer, because those hydrophobic tails pull towards each other. They have those hydrophobic interactions, and they pull towards each other to stay away from the polar environment that the water creates. So we're going to look at this picture again. This time I want you to label the area that is polar and the area that is nonpolar. I also want you to write where you would find the water in both of these, or where you would find the water in this diagram. So let's start with where we find the water. Here, there would be water. Let's say this is outside of the cell. But here there would be water. Down here there would be water. So we have our polar heads. And we have our non-polar tails. And then here in these heads would also be polar. So this region is polar, and this region is polar, and everything that falls in this area is polar, and everything that falls in this area is polar. But everything here is non-polar, which means it repels water. There's no water here. It's going to avoid the water. So that means that this part of this integral protein is non-polar. But the parts on the outside are polar. This peripheral protein, what do you think it might be? Right, it is polar because it is situated in the water environment or the polar environment. Now the cholesterol, what does it do? It acts for strength and fluidity. It keeps molecules from becoming stationary. It helps them move around, but it also helps hold them together. It provides the membrane with strength so that it doesn't fall apart. We tend to see more cholesterol in animal cell membranes than we have in plant cell membranes. We also see more cholesterol in animal cells in organisms that live in much colder environments for longer periods of time. The carbohydrates, they are the ID tags. So look around, you all have your ID tags on. You have those on so that we know you're supposed to be in Benton High School. If you had an airline ID tag, we would know you're not supposed to be here and we would have you leave. Carbohydrates are your ID tags. You have other cells in your body that read those ID tags as they walk around, like the teachers in the halls, and when you're walking into class, we're looking at your ID tags. There are cells that will read those carbohydrates and say, ooh, that ID tag says you're supposed to be in the body. But if they read an ID tag and there's an airline ID tag in your body, then it's gonna kick that cell out. It says it's not supposed to be here. It's a foreign invader. It's gonna kick it out. It's gonna react. Your immune system is gonna kick into defense mode to protect you from that foreign invader. That's why if you get organ transplant, you take anti-rejection drugs. Basically, it weakens your immune system, but it's keeping your immune system from rejecting those cells of that new organ. Then we have the proteins. We said there are peripheral proteins and integral proteins. Now these proteins, the transport and the carrier proteins, if you look at the picture here, they are integral. They go all the way through the membrane. But what do transport and carrier proteins do? They carry molecules through the membrane. They create a hole for molecules that can't go through the phospholipid bilayer. Some can easily come through the phospholipid bilayer, but a lot of them can't. They're too big, or maybe they have too much of a charge and they can't go through. So they have to have another way. And transport proteins, they can also be called channel proteins because they create a tunnel for molecules to go through. We also have what are called carrier proteins, where the molecule will enter. It'll in the middle, and then the protein will change shape, allowing this molecule to go out. That is a carrier protein. But what is their function? To move molecules in and out of the cell. And this is just a little short video. I'll show y'all real quick. Maybe I'll show you. Let's see. Nope, video is not working today, so try one more time. There we go.
There are two main types of transport proteins, channel proteins and carrier proteins. Channel proteins are tube-like and provide openings through which small, dissolved particles, especially ions, can diffuse. In general, each type of carrier protein has a shape that fits a specific molecule or ion. When the proper molecule binds with the protein, it changes shape and moves the molecule across the membrane.
All right, so it just kind of gave you a video of what they do so you could see them in action. Other proteins can have function or can function as glycoproteins. Remember, these are the proteins that have a carbohydrate attached to them. They function in cell-to-cell recognition. So here you can see, here's the carbohydrate, that ID tag. Now it is attached to a protein, so it's called a glycoprotein, but here's that ID tag, and this is one of the cells coming by. Here's its membrane, and it has a protein. This is called a receptor protein. But it is receiving that ID tag, and that ID tag fits, so this cell is supposed to be here. These proteins, these glycoproteins, can also be called marker proteins. They are marking the cell for what organism they're supposed to be in. Here, this is the same idea. It's just showing you, here's the cell, here's that carbohydrate, here's the receptor protein reading that carbohydrate to make sure that it is supposed to be in the cell. It's called cell-to-cell recognition. One cell is recognizing another one as local, as a good cell. It's supposed to be in the body. But these carbohydrates are very important for that. If those carbohydrates are wrong, your body will reject those cells. We also have the receptor proteins, like we just talked about. Some of them are used to recognize those carbohydrates, those ID tags. Other ones receive signals. So here you can see this receptor protein here. And you can see this signal is going to go fit in there. When that signal is received, it triggers this protein to start a reaction inside the cell. What is that reaction? It could be all sorts of things. It could be a signal that says, hey, this hurts. And when it says this hurts, that signal lands in that protein, which triggers a reaction in your neuron that causes you to send that message to your brain and back so that your reflexes will pull your hand away or whatever, wherever you're hurting, or so that you feel pain. So those receptor proteins receive a signal that cause a reaction within the cell. So if you look here, here's the signal, here's a receptor protein. This one is not in the membrane, but the signal hits the receptor. When it hits the receptor, we see this protein break off. This travels into the nucleus and it starts a response. So a receptor protein receives a signal and triggers a response. We do see those in the membrane. It's really important for those to be in the membrane. The receptor protein, remember, receives a signal, sends that, and that signal triggers a response within the cell. All right, and we said that this plasma membrane is selectively permeable. It controls what materials can pass through the membrane, which materials can't, and by doing that is how it's maintaining homeostasis. Remember, homeostasis is a stable internal environment. For it to be selectively permeable, for it to be picky, it has to have multiple ways for different molecules to come through. Some molecules can come straight through the lipid bilayer. So we have some molecules come straight through the lipid bilayer. And if you remember in the soap lab, the things that could go straight through the membrane were the things that were covered in soap, the things that were like the soap membrane. So here, the things that we see go through the lipid bilayer are lipid-soluble molecules, molecules that are similar to lipids. Non-polar molecules tend to go through the lipid bilayer. Other things that can go through the lipid bilayer are very small molecules, but they tend to be non-polar. Molecules that can't go through the lipid bilayer tend to be polar. They tend to be larger, and they need another method to get through. They need that channel like we made with the string in the soap lab. So those molecules will go through the protein, through this channel, because they can't get through here. If they try to go through here, they bounce off. But if they go through this protein that is specific for that molecule, it can travel right on through, just like it's supposed to. And tomorrow we're going to spend more time learning how and when those molecules move through. But just know that some go through the bilayer, some go through proteins. All right, so now at the end of your notes, there is a diagram and directions that tell you how to draw a cell membrane. It says cell membrane activity. Read through it. It's going to talk you through the same stuff we talked about with the hydrophilic head, the hydrophobic tail, and it shows you some drawings for different types of proteins, glycoproteins, and then it gives you directions for what you need to draw. But what you're going to do is you have this circle and you're going to create a phospholipid bilayer. So let's try one more time, maybe. So on your diagram, you're going to create a phospholipid bilayer, and there shouldn't be any gaps. You should have phospholipids filling up the entire circle, but it also tells you to put other types of molecules in there. So you might put in a carrier protein, and it shows you that a carrier protein looks like this. But I'm going to ask you to use different colors for different molecules. So we'll make our carrier protein blue. Maybe. And then we'll draw some more phospholipids. And then we're going to add some other molecules, and it tells you what to add and how many to add. But when you're done, you should have 2 entire layers of phospholipids with proteins, cholesterol, carrier proteins, transport proteins, peripheral proteins, all of those things added to your drawing. So that's what you're going to do. You have 15 minutes. Use different colors, use highlighters, different colored pens, whatever you need to, but you have 15 minutes to do that. All right, guys. So now that you have your drawing, we are going to use that tomorrow. Your drawing might look something like this. Now, obviously, this one is not hand-drawn, but just to give you an idea of different proteins, carbohydrates. Also, I need you to label where the cytoplasm, where the water is. But we're going to use this tomorrow as we start talking about different types of transport. And we'll revisit this tomorrow when we talk about evidence from the bubble lab and how this works. But this was from the bubble lab. Those of you at home, you do not have to fill this in. You didn't do the bubble lab, and that is completely okay. So you're not going to do this part. The front part you needed to do, but this part you just ignore. So my people at home, you can just put an X over this unless you want to ask somebody to let you see theirs, but you're not responsible for that. We're covering all the material in other ways as well, so you're going to be fine. Reminder one more time, if you didn't finish your drawing, you need to finish that tonight. 15 and 16 homework. It's due tomorrow, and you have a quiz on Thursday and a quiz on Friday. Guys, I hope y'all have a great day. If you'll pack up and stand by your desk while I clean, I will see y'all tomorrow.